Hello everybody and welcome to our Wednesday, February 7th lecture. So today we're going to be finishing up some of the components from chapter 20. We're going to primarily focus on chapter 20.2 and then again, as we have a little bit of time at the end of the lecture, sprinkle in some of these other concepts as we can get to them. So I'm going to go ahead and remind you, 5-point opportunity today. Don't forget to do that once this lecture is over. And then also make sure that you are prepared for the exam. So Friday you're going to need to turn in your homework for this chapter. And you are also going to need to make sure that you're showing up to lecture for the first exam. If you don't show up, then you don't get to take the exam. So make sure you are there. Here's our reminder of our objectives. So we've already covered our types of blood vessels. We've talked about the structure of each type and the route that blood can take through them. We've also looked already at objective 3 on here, which is our capillary exchange, and we've talked about the specific topic of venous blood return mechanisms. So today what we're going to primarily be looking at is blood pressure, resistance, and flow, and how all of these factors play in and depend on each other. And then again, if we have time, we'll look at some of these other ideas about blood perfusion to the brain and blood running through that pulmonary circuit. A couple of terms that we defined quickly at the end of yesterday's lecture, so we'll go back through them now. So blood flow, as a reminder, is going to be the amount of blood that is going to an organ or tissue or a blood vessel at a specific time. Okay, so we can compare that to perfusion, which is going to be the flow per given volume or mass of tissue. So we measure that as milliliters per minute per gram. So we may have a large organ that will have less perfusion but greater flow than a small organ. Okay, so if we think about the heart, which is about the size of a fist, and we think about the liver, which is going to be much larger than that, so probably 3 or 4 fists put together. The heart is going to have greater flow, right, because it's constantly getting a big supply of blood, but the perfusion may be less, because when we look at that flow rate per gram, the liver is so much larger that it is going to have kind of reduced perfusion compared to the heart. All right, and then, sorry for this last point, total flow is going to be consistent. So again, if your cardiac output is going to be 5 liters per minute, then you're going to have 5 liters of blood flowing through all of your organs during that given time. Your cardiac output can vary from minute to minute, so we can see a change in the total flow from time to time, but total flow is always going to be equal to cardiac output. So now we're going to talk about hemodynamics. So this is actually a physical principle of blood flow, and it's based on the idea of pressure and resistance. So what we're seeing down here is an equation. Let me point this out. So we have an equation right here where F is equal to flow. Delta P is going to be the difference in pressure between any two areas, and R is going to be resistance. So what the equation reads is that flow is proportionate to a change in pressure over resistance. So if we kind of think about what these different variables mean, flow is simply that. Flow is going to be milliliters per minute, so how much blood flow is going through a particular organ. The difference in pressure is going to be the difference in pressure between 2 points. So remember that blood is going to flow down a pressure gradient from an area of high pressure to an area of low pressure. If that pressure difference is great, so if we're going from an area of high pressure that's at 100 millimeters of mercury to an area of low pressure that's down at 20 millimeters of mercury, we're going to get lots of flow towards that lower pressure region. On the other hand, if our resistance variable is high, okay, so if something is pushing against that blood flow, is preventing the blood flow from happening, then we're going to get high resistance and the flow is going to be reduced. Okay, so let's look at this equation again. Flow is going to be proportionate to change in pressure over resistance. So if pressure is large, then we're going to get really large amounts of flow. If our resistance variable is large, so if we make this denominator big, that's going to reduce flow. Okay, so let's take a look at kind of this idea of pressure and resistance independently, and then we can come back and couple them together to look at this concept again. Talking about pressure, we measure pressure by determining the force that the blood is exerting on a vessel wall. So generally, we measure blood pressure at the brachial artery. So when you go to the doctor, they put the sphygmomanometer, that's a kind of a hard word to say, this big guy, on your upper arm, right? So they put it just above your elbow. That is going to be where the brachial artery is found. So the brachial artery is fairly close to the skin, and so we can measure the pressure of the blood moving through that artery. That artery is also relatively close to the heart. Okay, so at the heart is where we're going to have, at that aorta is where we're going to have the highest blood pressure. So we want to measure blood pressure as close to the aorta as we can to get a true value or a true reading of how much pressure is being exerted on the artery walls. So for clinical purposes, we're going to use that brachial artery, and we're going to record two different pressures, right? So when you go to the doctor, they give you a blank over blank number. The two numbers that they're giving you are the systolic pressure, so that's going to be the first number that they tell you. The second number is called diastolic pressure, okay? And you should kind of recognize these words, right? So systolic is going to be the peak arterial blood pressure that is taken during ventricular contraction. So during ventricular systole is where we're going to get that really big peak blood pressure number. The diastolic volume is going to be the minimal arterial blood pressure that's taken during ventricular diastole or between heartbeats. Okay, so systolic pressure is when the ventricles are actively contracting, actively pushing blood out through the aorta. That's going to add blood volume to the aorta. The aorta is going to have to expand, right? So we have those elastic tissues that are expanding to accommodate this volume of blood, and that is going to result in an elevated pressure within that artery. And then the diastolic pressure, again, is going to be the pressure in the arteries during diastole. So once we have relaxation of the ventricles and we're no longer putting as much effort into moving blood out through those big arteries. So in a healthy adult, normal blood pressure would be something in the range of 120/70, and this is measured in millimeters of mercury. There are additional types of blood pressure measurements that can be taken and used for different purposes. So one of them is called the pulse pressure. The pulse pressure is fairly simple in terms of calculation. So what we're looking at here is the difference between the peak systolic and the lowest diastolic pressure. So if your blood pressure was 120/80, you would take your systolic pressure of 120, you would subtract your diastolic pressure of 80, and you would have a pulse pressure of 40 millimeters of mercury. So this is going to be an important measurement in driving circulation and looking at the stress that's being exerted on kind of the smallest arteries by these pressure surges. Okay, so if we want to think about what's going on kind of downstream in our smaller arteries, when the heart is contracting or how much stress is on those small arteries during that time, we can look at our pulse pressure. The other thing that we can look at is the mean arterial pressure, which is the mean pressure that you would get by taking measurements at several intervals during one cardiac cycle. Okay, so this is going to be one cardiac cycle, and what we see is we have a big peak here, we then have kind of a dip off, and then we have a nice gradual slope to our diastolic pressure. So in order to calculate this, we can't just take an average of our diastolic and systolic pressure. We actually take our diastolic pressure and then we add to it about 1/3 of our systolic pressure. And the reason that we do it this way is because the low blood pressure portion is going to last longer than the high blood pressure portion. Okay, so we can't just kind of average the two, because that over represents the systolic pressure and under represents the diastolic pressure. All right, so those are just a couple of additional types of measurements that we can get when we use blood pressure. Kind of touched on this idea, but I wanted to solidify it. Blood pressure is going to change across the vessel types. So blood pressure is going to be highest in the arteries, okay, and it's very what's called pulsatile. And that means that we get big surges of pressure and big drops in pressure. Okay, so we get surges from about 80, which is going to be our low, all the way up to 120, and then it's going to drop back down, and it's going to surge back up and drop back down. So in our largest arteries, so this area right here is showing you what's going on in the aorta, so right outside of the heart, over and over, we're going to drop up. With each heartbeat, we're going to go down, up, down, up, down, up. So it's very pulsatile. Up and down, up and down. This is because the blood is coming out of the heart intermittently and only when the ventricles are contracting. So we get big spurts of blood volume when the heart contracts, and then it stops. So we get a big pressure surge, and then it drops back down. And then big pressure surge, and then it drops back down. As we move away from the heart, which is where we're going to have the peak pressure, we see that the pressure overall decreases. So as we move into the large arteries, we see a decrease in systolic and diastolic pressure. Both of those are going down. We also see a reduction in the pulse pressure, right? So the difference between the two readings is going to start decreasing. So here we're at about 100 over maybe 78. So it's going to be 22 millimeters of mercury difference. We're up here, we're at 40 millimeters of mercury. So we start to see less kind of big ups and downs in the pressure, and that's because we have the elastic and smooth muscle in the arteries that are trying to equalize the pressures between diastole and systole. So we start to even out those pressures as we move away from the heart, which is overall going to put less pressure and less stress on the walls of the blood vessels. So every time the heart beats, that puts a lot of stress on the walls of the large arteries. As we move down towards the arterioles and the capillaries and we reduce that wall structure, we can't continue to put these big surges of pressure on those vessels, because that would cause an expansion and it would really damage the walls and prevent them from contracting back down to the size that we need them to be. So we're going to see a continued decrease in pressure as we move to the small arteries. Again, much lower pulse pressure here. So maybe down into the 10 millimeters of mercury difference between a contracting heart and a relaxing heart. In the arterioles, we begin to see virtually no difference at the end, so maybe very tiny pulses between diastolic and systolic readings. By the time we enter in the capillaries, we've reduced blood pressure quite substantially, so we're down into the kind of 20 to 30 millimeter of mercury range, and we see no difference between when the heart is contracting or relaxing. So now we have a steady flow of blood as opposed to a pulsating or surging flow of blood. In the venules, small veins and large veins, we see a little bit more drop off in blood pressure, but we kind of stay in this 10 to 15 millimeters of mercury range until we hit the vena cava, and then we dip off as we enter back into the heart. Chart, fairly common, you may have seen it before, that looks at all of the different blood pressure categories. Okay, so if you have a normal or healthy blood pressure, your systolic pressure, so that's the number that they give you first, is going to be somewhere between 80 and 120, and your diastolic is going to be between 60 and 80. Okay, so you could be anywhere from 80 over 60 to 120 over 80, and that's a good, normal, healthy blood pressure. Hypotension is when you have low blood pressure. Okay, so when you have low blood pressure, you often you may have low blood volume, you may be kind of lightheaded and dizzy and have these kind of fatigue-like symptoms. You're going to have a systolic pressure of less than 80 or a diastolic pressure of less than 60. We then, if we go over our values, we enter into prehypertension or hypertension. So these are going to be our categories where we're going to be putting too much pressure on the arteries, which is going to cause them to weaken. We're going to have aneurysms. We're going to promote kind of these plaquey buildups just because the blood vessels can't handle everything that's happening. So if you're over 120 or over 80, you're going to be in one of these prehypertension or hypertension categories. And anything that's greater than 180 over 110 is going to be an emergency situation. Okay, so you don't ever want to enter into that highest category. There are factors that play into what your blood pressure is. So some things that can result in blood pressure increase are going to be our cardiac output, blood volume, and resistance to flow. Okay, so if we look here, if your blood volume increases, so we're actually adding more fluid to the blood, that just means that you have more blood present. That blood has to be somewhere, and your blood is almost always going to be in your blood vessels. So more volume equals more pressure on the walls, equals an increase in blood pressure. As your heart rate increases, the cardiac output increases. Okay, cardiac output, that's going to, again, put more pressure on those arteries, so we see an increase. And if the stroke volume increases, again, we're basically adding excess volume to those arteries, so that also causes blood pressure increase. The two factors down here are a little bit different, so these are mostly related to the heart or the blood itself. This one is blood viscosity. So you can think of blood viscosity as being how thick the blood is, right? We talked about blood viscosity back in chapter 18. Blood viscosity is going to be heavily impacted by red blood cell count. So red blood cells make up the largest proportion of formed elements. When we have excess red blood cells, we're going to have more formed elements and therefore kind of thicker blood. Okay, so thicker blood is going to be more difficult to move through a blood vessel than thin blood, right? Think about sucking up honey through a straw versus sucking up water through a straw. Okay, so that's what happens when you increase viscosity. We also can have peripheral resistance that can increase. So resistance is going to be changing of the vessel radius or changing of the vessel length. So you can kind of think of this as if we have excess friction kind of downstream that works against blood flow, and that is also going to result in an increased blood pressure. So, again, peripheral resistance is anything that opposes blood flow. Three things that can impact this, which is what I just said, are going to be blood viscosity, so how thick the blood is. And again, the primary factor affecting viscosity is going to be red blood cell count, but we also have those proteins. So if plasma protein concentration also is elevated, that's another factor that's going to make the blood thicker. For vessel length, think about the fact that the further the blood has to travel through a tube, the more likely it is to kind of accumulate friction. So even though we have these very smooth blood vessels through which blood should flow without problem, there's always going to be a tiny amount of friction that is accumulated. So if the blood is flowing from the heart to the lungs, that's a very short distance. It's not going to accumulate much friction. But if the blood is flowing from the heart to the big toe and back, that's the longest distance that it could possibly travel, and so over the course of that entire pathway, it's going to pick up more and more friction as it goes. And then vessel radius, and I want to point out this one in particular. This one is going to have a very, very big effect on on resistance. So now that I've virtually crossed it out there, but this is really important. So vessel radius is going to have the most influence on peripheral resistance. So over here in this image, we're looking at the difference that radius causes in terms of how much flow is occurring through a vessel. So if we start with a pressure of 100 millimeters of mercury, so we're coming into each of these blood vessels with the same pressure. If the diameter of the blood vessel is 1 millimeter, the amount of fluid or the amount of blood that's going to come out the other end is 1 milliliter per minute. Okay. If we double the diameter to 2, we have a 16-fold increase in the flow. Okay, so we go from 1 milliliter per minute to 16 milliliters per minute just by changing the diameter of our tube from 1 millimeter to 2 millimeters. If we double it again to 4 millimeters, we now see an increase in flow up to 256 milliliters per minute. Okay, so that is going to be a huge increase in flow rate because we have reduced the resistance as we increase the vessel radius. All right, so vessel radius is going to be very important in determining peripheral resistance. So what is going to change vessel radius? The primary thing are going to be our vasoreflexes. So vasoreflexes are going to be either constriction or dilation. Vasoconstriction is going to be when the smooth muscle of the tunica media is contracting. Okay, so the smooth muscle in the tunica media of the arteries or the veins is contracting, so it's shortening those muscle fibers and reducing the radius of the vessel. Vasodilation is going to be the opposite of that. So when the smooth muscle cells relax, that is going to cause the tube to dilate or open up wider. So it's going to have a wider radius. And so, again, these are just a couple of kind of side points here. We do see laminar flow in the blood vessels. So the blood that's in the exact middle of this vessel is going to flow faster than the blood that's out towards the edge. So this blood is going to be rubbing against the edge of the tube, so it's going to encounter that friction, whereas this blood that's right in the center is not going to encounter any friction. Okay, so we see this kind of laminar flow pattern, and this is an equation that's explaining why small changes in vessel radius result in large changes in flow, which is what we saw on the last slide. And so it should make sense, given what we're talking about with peripheral resistance, that as we move from the aorta towards the capillaries, we're going to see a decrease in blood velocity. So the speed that the blood is moving is going to decrease from aorta to capillaries. It then increases again as we move from the capillaries back into the vena cava. So we have lots of friction that builds up over time from the aorta to the capillaries, and we're also decreasing the diameter of the tubes as we go. So we start with our big thick arteries, we move to smaller and smaller arteries until we get to the capillaries, which are very narrow. So we're changing the radius of those tubes to become very, very small. The opposite happens when we're coming back into the venous system. So we don't have very much resistance in the veins, right? We kind of have these tubes that open very easily and they don't fight back, right? They don't contract or they don't split. And we're also merging or pooling large amounts of blood into one tube as opposed to having small amounts of blood in many, many tubes. So even though we do see an increase in velocity in the venous system as we're heading back towards the heart, the blood in the veins are never going to be moving as quickly as the blood in the arteries. So the arteries have so much pressure behind them that we still see this very quick movement compared to what's going on in the veins. We're going to see the arterioles playing a huge role in peripheral resistance and kind of our velocity. And so most of this is that we are really increasing the number of tubes that the blood is moving through, and we have a lot of control mechanisms in the arterioles. So the arterioles, they still have that smooth muscle. They can still kind of close off or open up and allow the blood to slow down as it moves into the capillaries or move it from capillary bed to capillary bed. And they are very muscular in proportion to their diameter. So what that means is that they're able to change their own radius quite substantially. So they can crank down, they can contract that smooth muscle and really reduce the size of the actual blood vessel, or they can relax and dilate back open, so they have these nice big tubes that blood can easily move through. So even though arterioles are just a small portion of the total blood vessels, they produce half of the total resistance. So they are really going to be in charge of slowing down the blood as it's moving towards the capillaries. And so these are just a couple of charts that are looking at what's going on in different aspects. So up here, this one's looking at vessel diameter. Okay. And this is looking at, I believe, velocity. So we see that as we move in the arteries, the diameter decreases. Okay, it's very small down here, then increases, right? I'm sorry, this is just vessel diameter. So our arteries are going to be large. They get smaller as they go down to the capillaries. We then see that the veins are actually even larger in diameter compared to the arteries. As far as overall area goes, the arteries don't have much area, but we see tons of cross-sectional area in the arterioles and the capillaries because we have millions of capillaries that are spread out over a huge area compared to just a few of these elastic arteries in one or two vena cavas. So these are very small proportionate areas, large cross-sections in these other blood vessel types. So overall average blood pressure, we already looked at this one, so it's high, and as we move away from the heart, it's going to decrease. And then velocity of the blood starts high, drops off rapidly, and pretty much bottoms out at the arterioles and the capillaries. It then does regain some velocity as it moves into the venous system, but again, we're never going to see the increase in the venous system to where we started in the arterial system. We're talking about peripheral resistance and kind of this idea of change in radius or diameter of our blood vessels, we should talk about how we actually regulate the vasoreflexes. So vasoreflexes are going to be these really quick and powerful ways to change blood pressure and flow. So we can very quickly increase our blood pressure or decrease our blood pressure using the smooth muscle in the blood vessels. Okay, so we can quickly squeeze or relax and really increase or decrease the blood pressure. So we have three ways that we can control the vasoreflexes. Okay, and I'm just going to briefly touch on each of these. So we have local control, neural control, and hormonal control. There are a lot of things going on with local control. So autoregulation is going to be when these tissues are capable of regulating their own blood supply. Okay, so we have this idea that when tissues are not getting enough perfusion, okay, so they're not getting a large enough volume of blood per minute per gram of tissue, what's going to happen is the metabolic wastes are going to accumulate. That is something that we don't want. So when the wastes accumulate, it actually stimulates vasodilation. Vasodilation is going to result in increased blood flow to that area. Okay. So to do this, we have some vasoactive chemicals. So these are substances that are secreted by any number of different cell types that can initiate these vasoresponses. And I am actually going to not to talk about these last 2 points on here. So we have some different components of this. What I really just want to point out or be clear on is that a tissue is capable of self-regulating. So it can regulate to a point, and it basically continuously monitors how much waste product, metabolic waste product, is present in the tissue. And when it reaches a point where it's too much, and we're kind of on that cusp of some sort of cellular damage, it can tell the surrounding blood vessels that they need to dilate and bring more blood into that tissue. We have central and autonomic nervous systems that exert control over blood vessel size. Okay, so this is going to be the brain telling usually system-wide areas that they need to either dilate or constrict. Most of the time, it's going to result in constriction of vessels. but it can sometimes dilate vessels, especially in the cardiac muscle when you are exercising. So constricting blood vessels is going to result in an increase in blood pressure, whereas dilating blood vessels is going to result in a decrease in blood pressure. Okay, so we can open them up a little bit more. So we do have 3 vasomotor centers that are going to regulate these reflexes. And so we've already mentioned a few of them. We have baroreflexes. So these are going to change blood pressure, right? So they're going to respond to increasing or decreasing blood pressure. These are generally short-term regulators of blood pressure. Okay, so your blood pressure can increase for a short period of time, and these reflexes will help bring it back down. If your blood pressure stays elevated long-term, these reflexes adjust to that new set point and no longer regulate for that. Regulation, which is going to be if you have a rapid change in posture or a sudden emotional spike, which is going to result in elevated blood pressure. If you have chronic hypertension, so again, your blood pressure is constantly elevated, the baroreflexes or the baroreceptors are going to just change their set point, and they won't be able to correct for kind of chronic conditions. So after a couple of days of chronic hypertension or hypotension, they just readjust, and so they no longer respond to that elevated blood pressure. So this is just kind of a flow chart that looks at what happens. With elevated blood pressure, we see our artery stretching. That's going to cause those baroreceptors to signal to the brain that we have an increase in blood pressure. So we have vasomotor centers which are going to be inhibited. They're going to reduce vasomotor tone, which means we're going to get dilation and blood pressure is going to drop. We also have cardioinhibitory neurons that are stimulated, which reduce heart rate. So if you reduce heart rate, you reduce cardiac output, which is also going to reduce blood pressure. Neural controls, which are going to regulate our chemoreflexes. So they are going to respond to changes in blood chemistry, especially pH. So pH is going to be the main driver of changing blood pressure for this purpose, but they're also going to measure oxygen and carbon dioxide. So again, I already kind of pointed out where some of these chemoreceptors are located. So we have them in the aortic and carotid arteries. And again, I'm going to move kind of quickly through some of this material. So if I'm not focusing on certain points, you can focus less on them when you're studying for the exam. But I just want to make sure that we're getting to some of these kind of bigger things. And so the next part of neural control is important for making sure that the brain is adequately perfused. So the brain constantly needs oxygen and glucose. It cannot function without those things. So anytime we have a drop in perfusion to the brain, we have a medullary ischemic reflex, which is going to increase blood pressure to the brain and make sure that we're maintaining oxygen and glucose supply to that particular region. That also can influence blood pressure. And so again, I'm going to move quickly through these because we're actually going to come back to hormones next week when we're talking about the endocrine system. So I'm just going to point them out here and then just move along. So angiotensin 2 we're actually going to talk about during the urinary system. So this is a potent vasoconstrictor. It's going to respond to drops in blood pressure or blood volume, and we're going to get system-wide constriction of the blood pressure, so we'll elevate or raise blood pressure kind of across the body. We have atrial natriuretic peptides, so these are actually going to be secreted by the heart. They reduce blood volume and promote vasodilation. Okay, so if they're reducing blood volume and promoting vasodilation, that's going to result in a reduced blood pressure. We have ADH, which is going to result in water being retained. When we retain water, we are going to elevate blood volume. Elevated blood volume also elevates blood pressure. And then epinephrine and norepinephrine, which you know as neurotransmitters, can also sometimes act as hormones. They are, in some cases, vasoconstrictors, in other cases, vasodilators, so they can be involved in both elevation and decrease of blood pressure. So overall, we can dilate and constrict our blood vessels, which gives us general control of raising blood pressure all over the body. So we can constrict or dilate all of the blood vessels, which is going to result in systemic or system-wide raising or lowering of the blood pressure. The other thing that we can use vasodilation and vasoconstriction for is moving blood from one part of the body to another. So this gives us system-wide control, but also local control. And so there are times, again, for example, during exercise, when we're going to reduce blood flow to the digestive tract and the kidneys, so we're not worried about breaking down food or producing urine when you are out running or out shoveling or raking or whatever it is you may be doing. And it's going to increase blood flow to skeletal muscles. Okay, so we're going to constrict the blood vessels that are leading to the kidneys and the digestive tract, and we're going to dilate blood vessels that are leading to the skeletal muscles. If you are sitting in your chair, you've just eaten dinner, you're going to dilate the blood vessels that are leading to your intestines and constrict those that are leading to your legs, because you're not currently using your legs, so you don't need a lot of blood flow to them, but you are actively digesting, so you're going to want increased blood flow to the digestive organs. Whereas if you are out being active, you're going to constrict, so vasoconstriction of the blood vessels that lead to the intestines and dilation of the blood vessels that are leading down to your legs or to that active part of your body. And this all goes back to which tissues are metabolically most active right now. And so we can compare where blood is being moved in the body when you're resting versus when you're out walking around. So when you're at rest, you're going to get large portions going to the digestive tract, to the kidneys, and to the brain. A little bit less going to cutaneous. Coronary is going to be fairly low, and then everything else. We have some going to the muscles. So we don't entirely shut off the muscular system, just reduce it. For moderate exercise, more than 70% of your blood is being moved to your muscles. We see much reduction in digestive and renal. Cerebral is going to stay fairly consistent because we always have to have blood going to our brain. More to the skin, a little bit more to the heart, but again, most of it going to the muscular system. We are kind of wrapping up the main ideas here, so I just have a few kind of little points to drop in for information's sake. We do have some special circulatory routes. One of them is going to the brain. So the brain is going to be very sensitive, and it is going to require that we have very little fluctuation in total brain flow, total blood flow to the brain. So regardless of what you're doing, you're always going to have more or less the same amount of blood being moved to the brain. So if we deprive the brain of blood for even just a few seconds, it results in loss of consciousness. And then within just a few minutes, so under 5 minutes, we have irreversible brain damage. So the brain is going to be very sensitive to lack of blood flow. So we get less fluctuation in blood flow to the brain than we get in any other organ. So the brain actually regulates its own blood flow. So it constantly monitors changes in blood pressure and changes in blood chemistry. And then we get responses in the cerebral arteries that are going to dilate or constrict as systemic blood pressure drops or rises. So your systemic blood pressure may drop suddenly as you stand up. We know when you start getting a little bit lightheaded after you stand after sitting down for a while. You're not going to see a similar drop in blood perfusion to the brain. Okay, so the systemic blood pressure can drop. Blood pressure to the brain is going to stay high because those cerebral arteries are going to constrict, or dilate rather, so we're going to get more blood flow to the brain. And then it's also going to regulate chemistry, right? So if we have changes in oxygen, carbon dioxide, or pH in the brain, those are going to trigger responses that will take over or control for that fluctuation. The circulation to the skeletal muscles is going to depend on the state of exertion. I think we have already covered this. It is highly variable, so unlike the brain where we have very little fluctuation, skeletal muscles we have tons and tons of fluctuation. At rest, again, most of the capillary beds serving the skeletal muscles are shut down. During exercise, they are open, and so we can increase blood flow tremendously. Excuse me. And so actually contraction is going to impede flow. So we see this kind of up and down, up and down, up and down, up and down. So this is when a muscle is actually contracting. So it's kind of interesting that we see this pattern here. And we also do have special circulation to the lungs. Okay, so during low pulmonary blood pressure, we're going to see slower flow in the lungs, okay, which is going to allow more time for gas exchange. And then if we're in a state of hypoxia, which is going to be where we are have low blood oxygen concentration, the pulmonary arteries are going to constrict and redirect to an area where we have better ventilation. Okay, so if there's a particular area in the lung that's diseased or damaged and oxygen is not reaching that area very well, we're actually going to shut off the arteries that go towards that area, and we're going to move the arteries towards an area that's actually receiving better ventilation, so receiving more oxygen so that the blood can pick up more oxygen at that location. We unfortunately don't have a lot of time in lecture to get into kind of all of the different blood vessels, so we've mentioned a few and we'll continue to talk about some of them as they relate to particular organ regions, but unfortunately we don't have time to go through which blood vessels flow to exactly which location in the body. I will kind of go through the anatomy of the pulmonary circuit at this point because it's kind of short, so we don't have to worry too much about it. But as we come out of the heart, so here we are in our right ventricle, we're going to come up through this pulmonary semilunar valve and into the pulmonary trunk. So this is kind of a shared artery that is then split into the left pulmonary artery and the right pulmonary artery. So these the biggest veins that are here, the biggest arteries that are here are going to be the pulmonary arteries. Once we enter into the lung, we actually start this branching process. So we can follow that this is going to branch into 3 different branches. Okay, so each of those branches is going to serve a different lobe. So we have our superior lobar artery, our middle lobar artery, and then our inferior lobar artery down here. And then we continue to split into additional lobar branches. So when we go through lung anatomy, or the bronchial tree anatomy, we're going to see that we have a primary bronchi that splits into lobar and segmental and all this other stuff. And so this kind of lobar branches will make a little bit more sense, but eventually we're going to get down to the very distal ends of these blood vessels. Those are going to form capillary beds that are associated with the respiratory membranes in the lungs. That's where we're going to have exchange of carbon dioxide for oxygen, and then we will follow our venous return backwards. And so again, the primary thing that is really unique about our lung blood vessels is that the veins are actually carrying deoxygenated blood, and the arteries are carrying deoxygenated blood, and the veins are carrying oxygenated blood. Okay, so that's different from what we see around the rest of the body, where the arteries carry oxygenated blood and the veins carry deoxygenated blood. At the respiratory membranes, okay, so these are called alveoli. They are these sacs that are full of air, so they're at the very end of the bronchial tubes, and they are closely associated with these capillary beds. So each of these kind of little bubble-like sacs is going to have its own capillary bed. So we have tons and tons of capillary surface area here, so we get lots and lots of exchange of those blood gases and those metabolic wastes and nutrients. And so you can see here we come in a pulmonary artery. We then go into, let's see, go this way. This becomes a capillary bed, so we go from deoxygenated to oxygenated, which again is going to be opposite what we see in all the other body tissues. That oxygenated blood is then going to be carried back out and up into this pulmonary vein and be returned to the left atrium. So we can bring in the anatomy of the heart into the pulmonary circuit when we're looking at the blood vessels that are involved as well. All that we're going to talk about for chapter 20. And I will say again that anything that I didn't talk about in great detail, so if I skipped over a couple of points that were on the slides, that information will be represented proportionally on the exam to the amount that I talked about it. So focus really heavily on the stuff that we spent a lot of time talking about, right? We talked a lot about our different blood vessels. We talked about their structure. We talked about how their structure is associated with their overall function. Okay, so think about the function of the arteries, think about the function of the capillaries, think about the function of the veins, and how the structure of those is directly associated to what they're actively doing in the tissues. Don't spend so much time worrying about autoregulation of the tissues or the hormonal control component. So those are going to come up again later, and so we'll touch on them a little bit more heavily. That doesn't mean to ignore large parts of the lecture or lots of the information from the textbook, but just concentrate your studying on areas where we spent lots of time talking about it. So the first exam is going to be Friday. Okay, so that should be 2 days from today. You can go back and listen to all of the lectures from the last 2 weeks. Look at your notes from the lectures from week 1. If you have questions that you need answered, you can discuss those with Vlad. You can also discuss them with a tutor. So if you need to set up a tutor through the tutoring center, they are excellent and they will be very helpful. And make sure that you're completing the homeworks, using any supplemental resources that you need, and getting everything solidified in your mind as it will help you on the exam. So with that, you will have a 5-point opportunity available to you at the end of this lecture. So it'll open at noon. And then it's due tomorrow at 11 a.m. So please, again, make sure you're completing those. There will be no 5-point opportunity on Friday. So since there is an exam, there's not going to be any 5-point opportunity. So for this week, you will just have 5-point opportunities on Monday, Tuesday, and Wednesday. Also, don't forget to turn in your homework for this chapter. So you want to do that when you come to the exam on Friday. Okay. And so that brings us to the end of week 3. Remember, exam 1 is going to be on chapters 18, 19, and 20. So you will need to make sure that you are reading those chapters in your textbooks in order to be prepared for the exam, listening to the lectures, and going to the review sessions. So good luck on Friday, and I will be seeing you all in just a few short weeks.